So I just wanted to thank the organisers for giving me this opportunity to speak. Today I'll be talking about Intron signal that's taken from RNA-seq data. So when we're talking about gene expression analyses using RNA-seq data, we generally focus on summarising the reads that we get from the experiment into genes. Specifically, this would mean counting the reads to annotated exons. So here I've got this little schematic of what an annotated gene may look like. And in green, you've got the annotated exons and you've got the reads on top of it. So only the reads that overlap an annotated exon gets counted. So for this gene, we would say that it has a gene count of 10. So gene counts, what we normally do is compare the gene counts from, say, two different conditions, group A versus group B. And for example, if group A has a higher count than group B, then we'll say that group A is more highly expressed than group B. So hopefully these are things that most of you guys, probably know quite a bit about, but I just sort of wanted to set the background of it. I wanted to point out that typically only exon reads are actually considered because the assumption is what we get from the RNA-seq experiment are mostly mature mRNA transcripts, and they're the ones that are generally of interest. So whereby a mature mRNA transcript should have the intronic region spliced out. So everything that's in the orange bits, you sort of don't really expect to see much of that going on. Two of the most popular library preparation protocols are your polyase selection and your total RNA selection with ribodepletion. And both of those are enriched for these mRNAs that we're interested in. So, all of that kind of checks out if that's the assumption that we're working off. So the motivation for the work that I'm presenting today, which is also part of a publication that was like a very recent publication, I think last month. But the motivation of the work comes from a previous paper we published in the lab. So it's the one down the bottom with the RNA-seq mixology paper in the Nucleic Acids Research Journal, where one of the figures showed that intronic, so intron counts that are summarized over those annotated regions, if we take a log fold change comparing two different groups, where in this case, the two different groups are human lung adenocarcinoma cell lines. So if we compare the intron log fold changes in the plot on the left with those that are the exon log fold changes, you'll see that there's a strong positive correlation. The plot next to it just shows what the correlation would look like if you're looking at the gene-wise exon log fold changes versus the intergenic region that is adjacent to it. So this was quite a surprise to me because I've been doing RNA-seq analysis for quite some time, and I've always just been doing the typical thing where I look at the exon counts, and I've completely ignored what's happened in the introns. So it was a bit of a surprise to me, so I wanted to look a bit further into this to see what was going on. So I thought, all right, that's really simple. I was really excited. I thought, great, I can make use of some of these intron counts, maybe put it into a differential expression analysis and get some free information out of there. So I was like, let's make use of the intron counts. But I had no idea what they actually represented or how I could use them. Like, if you don't know what they represent, then don't really suggest that you just, get really gung-ho about using them. So I thought, all right, I'll look into the literature and surely someone's got an answer of what these intron reads represent. I thought it could be quite straightforward. So here I've listed a few publications. Firstly, I saw that some papers mentioned that intron reads could represent noise, experimental and transcriptional noise. There's other ones that claim that it's unusable. And then there are some publications that talked about how they can be used. So in particular, the third publication here talks about how it's correlated with nascent RNA. And then there's other publications that use intron reads to look at intron retention. So where does that kind of leave me? Well, some of this was really conflicting. In particular, even out of the papers that say you can make use of the intron reads with the publication on nascent RNA, it doesn't mention anything about intron retention, whereby the nascent RNA and the intron retention are very distinct RNA transcription processes. On the other hand, the papers that talk about intron retention generally assume that if you're working with poly-A RNA, then you're only getting mature or processed mRNAs. So that again conflicts with the nascent RNA study. So, there was no consensus between the publications, and I sort of, went back to square one and didn't really know what to do with that. So instead, I decided to do my own data exploration. It certainly did help to sort of see what other people had done and then do my own investigation into these reads. So I did want to point out that this is fairly data exploratory because I found part of the problem was in some of the previous publications, perhaps there was a biological signal that that group was interested in, and so they went down that path. But for me, it made it really difficult to just throw a random data set at any of these methods and sort of decide, well, which method should I apply? Should I assume it's noise? Should I assume it's nascent RNA, or should I assume it's intron retention? I sort of didn't know what to do with it. So I went back to square one to try and look at it myself. I looked at 3 different data sets. So there's 2 that's from human data and 1 that's from mouse. So the first data set is actually the data set that's taken from the earlier study that I pointed out of the lung adenocarcinoma cell lines. So I'm just going to call these the human cell lines for now. 2 different cell lines, and there's a total of 12 libraries where there are 3 replicates each of the 2 different cell lines done in poly-A protocol and also total RNA protocol. The thing to point out about this data set, because we use it a lot downstream, is that we prepare 3 replicates of, say, one of the cell lines, and then the same replicate is split into a poly-A or a total RNA. Well, it's split so that it's done with both. So we kind of consider these the same biological samples. It's just that they have different library preparation. I also have some human immune cells that are done on poly-A RNA libraries and some mouse memory cells that are done on also poly-A RNA libraries. So here I look at 2 different types of counts. So again, you get this same schematic of what an annotated gene looks like. We still count those reads that are in green. So my gene-wise exon counts, but additionally, we also count the intron counts. So here the intron count would be 3, whereas the exon count would be 10. These are summarized over genes. Something to notice is if you've got a read overlapping an exon-intron boundary, then they are considered an exon count rather than an intron count. The reason why we do this is because, well, then your exon counts represent what you would typically count, or the information you would typically use using RNA-seq data, whereas in orange with the intron counts, they're kind of the extra reads that people generally sort of just discard and don't do anything with. So I first look at, I guess, a very basic question. Are the intron reads prevalent? Do I get intron reads in all the samples, in different data sets, etc.? So the 3 different colors you see in these box plots are the 3 different data sets that are separated into their biological and technical groups. So for example, in purple, we've got the cell lines, the 2 different cell lines. The first 2 are the 2 different cell lines done with a poly-A protocol, followed by the 2 cell lines done in a total RNA protocol. So that's what the T represents there. And then you've got the other 2 data sets there as well. So one observation we make is that intron reads are prevalent, even if it's at fairly low rates. So this does range between 5% and 20% in terms of this plot. In the poly-A samples, this is roughly between 2% and 14% of reads in a total set can be considered intron reads. So this is roughly a mean of 7%. in the total RNA, this is 21%. So that's our first observation, intron reads are prevalent, even if it's at low or moderate rates. The second observation we make is that the intron read proportions appear fairly consistent within technical and biological groups. So you'll see that the box plots are fairly consistent within each of its own, I guess, like column that's presented there, but it can vary quite a bit between the different biological groups or technical groups. The third observation we make is that there are more reads in the total RNA protocol. So remember, if we're just focusing on those purple box plots, between the first two box plots and the next two box plots, the only difference is the library preparation. And there's quite a big jump there in terms of the intron reads. There's roughly 15% more intron reads that are in the total RNA when compared to the poly protocol. So you might think, even if we just focus on the poly samples, you might think, all right, this is a fairly low percent. It could be 2%, it could be 7%, 15%, etc. It's not that substantial, but actually the accumulated counts really add up. So if you're looking at a library size of roughly 30 million reads, which isn't a very large library size these days, then if we're using the mean percentages, you roughly get 2 million reads in the poly RNA libraries and 6 million reads using the total RNA libraries. So that information starts stacking up in the intron reads. This is the first plot I have there is the same one that you're looking at before, just flipped the other direction with the intron proportions. Just some extra plots you've got there are the exon proportions, and then a correlation between log intron counts and log exon counts. So just some additional things that we see is that the exon proportions are higher than intron proportions in a given library, which is what you would expect. So in other words, if we're looking at roughly 70% for the exon proportions, then you use roughly 70% of the RNA-seq data when you count the typical way. So this is a very rough statement of that. You also see that the exon proportions are lower in the total RNA. So again, looking at the purple box plots, there's that shift of proportions between the first two and the next two there. So there's roughly 20% less exons in the total RNA there. For the correlation, they roughly hover around the 60–70%. So exon, intron, log counts are positively correlated using Pearson's correlation. So some extra observations we've made, just that's very general about 3 different data sets and different groups that we have there. The next thing we do is to perform some unsupervised clustering using multi-dimensional scaling plots or MDS plots. So on the left are your intron MDS plots, and on the right are the exon MDS plots separated by the 3 different data sets. So each of the points that are on the plot represent a sample or a library, and the colors are meant to reflect their biological or technical groups. So you can see that the libraries cluster by their groups, whether it's technical or biological. And this is a really important observation because it shows that in an unsupervised manner that the intron reads are informative. It can distinguish the groups that we have, which is really, really important. And one other thing I wanted to point out is that if we just focus on the top plots, then you'll see that differences in intron reads are greater between library protocols than they are for the exon MDS. And what I mean by that is along dimension 1, you'll see that for the intron reads plot, red and blue is separated, and red and blue is separated, and the difference between those are the library protocols. So red is the poly cell line and blue is the total RNA cell line of the same cell line, and similarly with the green and purple. But if you then look at the exon plot, dimension 1 actually separates between the different cell lines in the same protocol. So green and red, where you have the 2 different cell lines in the poly A. So those intron differences are So those different transcriptional differences are bigger for the intron reads than they are in the exon reads. So we do start making some guesses as to what these intron reads represent now. What is the biological signal that it's picking up? So in the previous slide, I showed that the intron reads are informative. Now we sort of want to make some guesses as to, well, what signal is it picking up? Well, we see evidence that it is mostly pre-mRNA signal. So I've summarized it very briefly here in these slides, and obviously there's more detail about it in the paper. But just very quickly, I just wanted to sort of look at some of these points. Why we think it's pre-mRNA signal is that the majority of expressed multi-exonic genes, so genes that have some counts, that have more than one exon, express or have intron and exon signal. So that means most genes that have expression have both exon and intron expression, in other words. So this is by looking at the human cell lines. I've got asterisks next to like the next 3 points because these focus only on those genes that are expressed on intron and exon compartments. So we're looking at the plots on the left now. We see that there's a strong positive correlation between exon and intron counts. So this is something that we had showed previously, but particularly they look at the log CPM and the log RPKM here. So you see that there's a strong positive correlation when we summarize the counts in that way. Looking at a completely different data set now, we look at megakaryocytes and platelets. Why we look at megakaryocytes and platelets are that platelets are produced from these big megakaryocyte cells where platelets are anucleated. So they don't have a nucleus, which means we can expect that we won't see any pre-mRNAs if there is. So if we're assuming that most other cells that are nucleated have some of your processed and unprocessed signal from mRNAs, then the platelets that are anucleated should only have your mature mRNAs. So here you'll see in the plot in the corner that megakaryocytes do in fact have intron counts, whereas the platelets do not. Next to it is the plot that shows that if we're looking at the exonic regions, there is that positive correlation that we would expect. Last of all, we look at genes and their coverage patterns. And when we look at the coverage patterns of, again, the human cell lines, we'll see that, so these are the coverage patterns that are separated into short, regular, and long genes based on the length of the intronic region. And then we've got the 3 different replicates of the poly A and the total RNA represented by black and gray. So if we first focus on the total RNA signal, that then across the gene body from 5' to 3' end, that pattern is fairly uniform. If we look at a poly A protocol, then it tends to be higher on the 3' end. So if we purely look at this, we can sort of go, well, not sure which one to trust in terms of the different library protocols because they are meant to be the same biological sample, but the patterns show that it looks like the gene is unspliced or partially spliced in the intronic region. So that's where we're like, all right, let's just assume, and based on all that information that we see there, that intron reads are predominantly from pre-mRNAs. So what can we do with this signal that's from pre-mRNAs? What this means is the typical or classical DG analysis, differential gene expression analyses that we run on exon counts, already includes signal that's from the pre-mRNA because the pre-mRNA component will have those exon counts included. So what we propose is a method that complements the classical DG analyses and includes intron reads to measure those early transcriptional changes as well. We call this method index, which is meant to stand for intron differences to exons. And on the left, it's just a little schematic showing the workflow of how index works. So I'm not going to go into the full details of this. I just wanted to point out that you need gene-wise exon counts and gene-wise intron counts. And the general pipeline follows a limit-voom workflow. And then you get some index categories as output. So I did want to talk about the index categories. The index categories you get in that you can see in the table are, you've got your plus. If it's a plus category, it shows that it's differentially expressed in exons and going up. It's also differentially expressed in the intron counts and it's going up. If it's minus, then it's going down in both count sets. Exon plus means that it's only differentially expressed in the exons and going up, but not in the introns, etc. So there's a few different, I think 8 different categories there that sort of classify the direction of change as well as the significance of change based on a certain threshold. So here we've got the results from the index method for 2 different data sets, the human cell lines and the mouse immune cells. On the left, those bar plots reflect the number of genes that were placed in each group after we run the analysis. So they are ordered from the smallest to the largest group, and you'll see that most genes are differentially expressed in the same direction between the exon and intron counts. So those are the blue bars that you see that's popping up. The next biggest group are the exon-only groups. So the exon plus and the exon minus groups in the pink and red, followed by those orange groups that are the intron-only genes, and then a much smaller group as the mixed signal, so where it's differentially expressed in both sets but going in opposite directions. So when we look at the box plots, it's not so obvious for the mouse immune cells, but a bit more obvious for the human cell lines. I've also summarized the total intron length of the genes that were placed in each of those groups, and then they're ordered by the median intron length. What I notice is that for those exon-only genes, they tend to have a short total intron length. So that's the red box plot that you see on the left. And this is true of both data sets that we looked into. On the other hand, the intron-only genes tend to have longer intron lengths. And the mixed groups also have longer intron lengths. So I have a go at sort of interpreting, so these are the results, but let's have a go at interpreting what this might mean biologically. So looking at those genes that are in the plus minus groups, it looks like transcriptional changes are similar for both the intron and exon count sets, which would make sense if we expect exon counts to represent mature mRNAs as well as pre-mRNAs, given that we're saying that the intron counts represent pre-mRNAs. So there is that overlap and that could be expected. When looking at the exon-only genes, you can imagine, maybe it's not so obvious because I haven't mentioned it yet, but actually intron counts are fairly low, as you see as a general statement to data sets. You'll see that they're roughly 7% as opposed to 70%. So intron counts are generally low but long regions. So overall, if introns have low coverage and short regions, then it's unlikely for it to accumulate high counts, which means that when you're looking at those shorter genes, it lacks statistical power for testing. As you can imagine, the opposite is true. When we're looking at the intron-only genes, then even if there's very low coverage in those intronic regions, because it's got a very long region, it could accumulate quite high counts. This is particularly advantageous if we're looking at signal that's only in the pre-mRNAs, because we expect that the pre-mRNA signal to be still a fairly small proportion of the mature mRNAs that's in the data set. The mixed group is a fairly small group, so don't want to go through too many guesses of what's happening there, but biologically this could certainly happen where there's an increase in pre-mRNAs at the same time that there's a decrease in mRNAs, for example, in nonsense-mediated decay. What does this all mean? So it's possible that even the exon-only and the intron-only genes can be reclassified to the both group if we were sequencing at greater depths. And we do get a few more DE genes and an extra layer of information about the data. So just very quickly in the last maybe 2 minutes, I also wanted to talk about, well, what about intron retention? Intron retention was also one of the things that was mentioned as something that you can look at using intron counts, and I think this makes sense. So we also have this Superintronic software, which I'm not going to go into details for the interest of time, which looks at BAM files or aligned reads. And what we do is we select for genes that have a substantial number of intron bases that are much higher than other introns. So we still take into account that most of the intron signal comes from pre-mRNA. We don't just pick out any intron signal and say, well, that must be intron retention. For the human cell line data set, we come up with 43 genes that we select based on certain thresholds. And this becomes quite easy to manually check their coverage profiles. So here I've highlighted 3 of them that appear fairly intron retention-like. You'll see that there's a big bump in the orange or the intron coverage for a very particular region relative to the rest of the coverage in the area for these 3 genes. So out of the 43 genes, 36 of them appear fairly intron retention-like, and we can go through each one of these using our Superintronic plots. So here's a brief summary of all the things we went through. Intron reads are prevalent. They can provide signal to distinguish between groups. They're mostly from pre-mRNAs, but you can also find signal from intron retention. Some other considerations that I won't go into is whether there's incomplete gene annotations and that difference in intron signal between your total RNA and poly-A libraries. So on the left is a publication that we have, so feel free to look there if you feel that I didn't go through enough details or you want to know a bit more. Thank you for listening, and last of all, thank you so much for the organisers for doing this last minute switch because of it being such a funny year of making this a virtual event. You guys have done such a great job, and I look forward to collecting my event sticker in person when I get the chance to. I am happy to take questions.
Thank you, Dr. Law, for a great talk. So yeah, okay, I see a question. Can you read it or no?
Yeah, let me just switch out of my screen.
Feel free.
I don't have it. Hmm? I don't see the question. My last one is related to Marco.
So if not, I have a question for you. So what's going to happen if you combine both intron and exon, basically addition, like put together? Is it going to add a power, you think, to the differential gene expression analysis because you have more counts? This is the first question. I have to think about a second, I forgot actually. So go ahead and go that one.
Sure. No, that's a really great question. That's actually where I sort of thought to tackle this problem initially was just, let's just count in the gene body and get more counts and do a DE analysis. So I did try some of those strategies and found that there were times when some genes overlapped and times that genes didn't overlap. That's why we have now, instead of just taking the gene body, broken it into exon and intron counts. But after exploring it in using the index method, I do think it makes sense to also just combine them. I know people who have done that, especially for single cell data where they've noticed they've got a very high proportion of intron counts. They're already struggling with counts. So they're like, let's just, rather than getting no DE genes, if I count into the exons, let's count in the gene body and get a 40% increase, like for a particular data set that's poorly annotated.
So any question there? Okay, so then if not, I have a second question. So this is do you observe a lot of intron exon junction, like in proportion? Like, is this legit? If you have, you would assume the percentage of intron exon junction in terms of the number of reads, the proportion to the length of the intron, right? If it's longer one, then it means like intron exon junction proportional wise should be smaller.
Yes. That's not something we have looked at directly. So I think that's a really good approach. I think most intron retention methods look specifically at the exon intron junction to call it a retained intron, which I think is a really nice approach. Currently, part of the reason why we haven't looked at it specifically is because the variation in the intronic area is so variable that it's really, really hard to summarize. But I think it's something worth looking into.
Okay, thank you.